Hello. Today we're going to be talking about phototransduction. And where we are going to start is, what is phototransduction? So phototransduction is the process of converting light stimuli into electrical signals. This is done within the eye. The eye contains two different types of photoreceptors, the rods and the cones. Both of these are divided into three sections. You have the outer section, the inner segment, and the synaptic terminal or synaptic ending. Those can be seen on the right-hand image. The longer purple structure is the rod. The shorter green structure is the cone. Both of them are labeled their outer segment, inner segment, and synaptic ending. And the goal of these structures is to transform light energy into electrical signals that they're then able to be interpreted by the brain. Rods and cones. So light enters the eye and is focused to the retina. These rods and cones are extensions of the CNS. Rods provide indistinct gray vision at night. They have a higher sensitivity to light, and they are contained in higher concentration on the periphery of the retina. The cones provide sharp color vision during the day. They provide higher resolution. And then photoreceptors polarize on light absorption, which is different than other receptors that we've dealt with. Receptors typically are depolarizing when stimulated. So that is one of the unique things about photoreceptors. So the mechanism, there's three major components of the rod and the cone that are able to create these stimuli. You have we're going to follow the mechanism through the rod today, although it is the same for both. Cones just preferentially will absorb light in different parts of the visible spectrum. Both of them also have an opsin. In rods, when we're talking about the opsin, it is specifically known as rhodopsin. So light photon is going to come in and it's going to strike the rhodopsin. This is the first section to undergo transformation. In the rhodopsin, the cis retinol is converted and is no longer bound to the opsin. This opsin is then free to bind with an enzyme to convert GDP to GTP, which can be found on transducin, that second structure component that we're talking about. This unit from the transducin then is going to break away and will remove an alpha subunit from PDE, which is also known as phosphodiesterase. The alpha subunit is what was deactivating PDE. So once they are both removed, PDE will become active and will convert cyclic GMP to GMP. Normally in the cell, this cGMP gated sodium channels are held open, which causes sodium ions to rush into the cell. This is going to produce a continual release of neurotransmitter. Once this GMP is formed, however, the channels close, which will actually inhibit neurotransmitter from being released. This neurotransmitter was inhibiting the bipolar cells. So this neurotransmitter was inhibiting the bipolar cells. So once these bipolar cells become active, they will then start to transmit signal as well through. These bipolar cells do not have any axons. And then this picture on the right, before we go much farther, I just want to show, this is explaining kind of what I just referred to. So that light comes in, it first converts rhodopsin. From the rhodopsin, it's able to convert that GTP to GDP, which then is going to take that alpha subunit and convert PDE into an active form, which will convert cyclic GMP to GMP. These sodium channels were held open by the cyclic GMP. Once we are converting it to GMP, the sodium channels will close. So again, the bipolar cell is now releasing the neurotransmitter because it's not being inhibited anymore. This depolarizes the ganglion cells, which causes a burst of action potential along the axons, which is going to depolarize and create action potentials. So when we had our cones or our rods activating these bipolar cells don't actually have axons, so nothing is traveling down. There's no action potential that's moving throughout. When the cell depolarizes, that's what releases the neurotransmitters to those ganglions. And then the ganglions do have axons, so they depolarize and create action potentials. The signal then ends up getting down to the thalamus, specifically to the lateral geniculate, which is what's going to process those symbols, those signals to be processed by the brain. And then just to go over a few differences in the rods and the cones. Rods have these longer outer segments. They have many discs, which help to make them more sensitive to light, which is why they are actually better at detecting when it's darker out, or that sensitivity to being able to see light in dark areas. They have rhodopsin as their opsin. And then they also have many rods converging on a single ganglion. I'm just going to go back and show you in this picture what I mean by there. So these gray, these are our rods. And as you can see, they don't just talk to one bipolar cell. They're going to be talking to a few bipolar cells, which then are allowing them to talk to a few different ganglion here. And the point of that, what that does is that actually, you could have output from more than 100 rods converging via these bipolar cells on a single ganglion. So it's going to allow for an action potential to be created even when there's little stimuli coming in to stimulate that rod. So if only one rod was going to one bipolar cell, which went to one ganglion, when you only had a little bit of light come in, it wouldn't be enough to actually set off that action potential to be picked up by the brain. So when you have a few different rods that are all kind of going to that same ganglion to create that action potential, it allows there to be a readable stimulus that can be picked up by the brain. And then our cones. So our cones have these shorter outer segments. They contain specific they're going to be detecting specific wavelengths. So they're actually concerned with color. There is a private line connecting to one ganglion. So here you'll see for the cones, they're connecting to one to one bipolar cell, which connects to one ganglion, one bipolar cell that connects to one ganglion. And this is because we're not as concerned about the resolution, because these are meant for vision during the daytime and being able to differentiate colors and create better resolution. So since we know exactly which point this is coming from, since one ganglion goes to one cone instead of, multiple rods that go to one ganglion, we're able to pinpoint the location of where exactly that's coming, which creates this better resolution in the image that ends up getting processed by the brain. This also is providing our color vision, and there's different options that are located in each cone that specify whether they're picking up the wavelengths associated with red or with green or with blue, where our rods are just picking up and pushing on, that stimulus and that connection that it's seeing gray. And these are the references that were used for this presentation. Hope you learned a little bit about how your eyes connect with your brain to make phototransduction. Thank you.